Hello, everyone. Thank you for joining us today. I'm Erin Newbern, the moderator for today's webinar, and I'm very pleased to introduce Christelle Johnson, who will lead the webinar titled Accurate and Comprehensive Tumor Characterization Using Augmented NGS. Christelle is a member of our Personalis field application scientist team with several years of cancer research experience from her previous work at Harvard and Cornell. Following Christelle's presentation, we will have a Q&A session. So at any time during her talk, you have an opportunity to send questions that you'd like addressed. So please notice on your control panel there the ability to type in questions. This will be sent to the organizers for the Q&A time at the conclusion of Christelle's slides. So thank you again for the opportunity to speak with you today. I'd now like to welcome Christelle Johnson.
Thank you, Erin, for this introduction. And welcome everyone, and thank you for joining our webinar. Today I will be discussing assays that Personalis has developed for tumor profiling and characterization using an enhanced sequencing methodology. I will start with a brief overview of the company and our mission, followed by a description of our patented sequencing technology and how we differ from other services. Then I will introduce the different products that we've built to meet specific needs in cancer research and clinical trials. Before I get into more details about the products, I just wanted to briefly introduce who we are as a company and the breadth of assays that we perform. Personalis is a service provider for clinical and research next-generation sequencing. We offer solutions for Mendelian disease diagnostics, cancer services for research and clinical trials, and we also offer assays for complex disease and discovery research. And these products are carefully designed with corresponding informatics solutions in-house that are unique to Personalis and provide the most comprehensive and accurate platform. Personalis labs and offices are located in Menlo Park, California. The company was founded in 2011 with the mission of developing innovative, comprehensive, and high-accuracy sequencing and interpretation services for both cancer and inherited disease. We have a broad set of research and clinical services in these areas with customers in 12 different countries spanning pharma, biotech, hospitals and clinicians, government, as well as academic centers and researchers. The labs are also CAP accredited and CLIA certified, and all assays are performed following good clinical practice guidelines. We use Illumina instruments as our sequencing platforms, and we are able to handle different sample types from formalin-fixed, paraffin-embedded tissue, fresh frozen, bone marrow aspirates, peripheral blood mononuclear cells, whole blood, as well as other types of tumor samples. Our scientific founders are largely from Stanford School of Medicine and UCSF School of Medicine, and as we've grown as a company, we've taken on few additional advisors in areas of clinical informatics, genetics, and genomics. The management team also comes from various backgrounds and large institutions and has extensive experience in next-generation sequencing and bioinformatics. For the purpose of the webinar today, I will be discussing our cancer portfolio and the associated bioinformatic pipeline. But first, let's talk a little bit about some of the challenges in next-generation sequencing. In general, the next-generation sequencing process can be divided into 4 different components, and these include sample preparation and sequencing, alignment and variant calling, annotation, and analytics and interpretation. The challenges in the field are the different errors that can be introduced at every step in the process. For example, gaps and inconsistencies in the sequencing coverage, incomplete human reference genome, issues in the annotation databases, and all these really propagate downstream and lead to missed variants, inaccurate analytics, and poor results. In addition, as we moved into cancer, we've recognized that there is a lot into cancer that is really unique and doesn't apply to other areas of medical genetics and genomics. And this could range from the tumor samples that are formalin-fixed, paraffin-embedded, to small sample amounts, RNA DNA quality, to also the tumor heterogeneity and low allele frequency mutations in cancer. So we've put together a comprehensive list of all the different challenges in cancer and try to compensate for all of those to accomplish an accurate assay. And we developed a platform called ACE for Accuracy and Content Enhanced that provides augmented coverage, an accurate variant calling, and a comprehensive annotation solution to achieve better results. How does Personalis differ from other service providers? With a typical service, you'd be able to analyze a tumor DNA sample on perhaps a small size panel, 50 to 500 genes, obtain limited informatics solutions, limited annotation with a few databases, and at the end, a DNA report. These solutions do not meet the needs in the immuno-oncology area, but most importantly, they do not address the inaccuracies in sequencing. Personalis has gone beyond the typical options available and has capabilities in analyzing both the tumor DNA and the matched normal sample for the most accurate representation of the tumor biology. But also we have optimized extraction protocols to obtain RNA from that same tumor sample, important for integration analysis. All these configurations can be run on our extended cancer panel for DNA and RNA, which is the largest on the market and includes over 1,300 genes relevant to cancer, or on our augmented cancer exome and transcriptome platform that allow broad tumor profiling, and this is especially important for cancer immunotherapy needs. We also provide comprehensive informatics reporting and have strong annotation solution of over 40 different databases. We're also flexible with how we report the data to meet the needs of every project, in addition to providing access to all the raw files. In the next few slides, I will describe the principle of our patented exome augmentation approach and how we've developed an exome assay based on accuracy and uniformity in coverage. The biggest challenges in sequencing are really the gaps and inconsistencies with the standard exomes. For example, this is a sequencing plot of STK11 gene using the Standard Exome Agilent Clinical Research exome. The plot shows all exons concatenated on the x-axis and the read depth on the y-axis. And as you notice, although this gene is sequenced at high depth, in this case at an average of greater than 100x, it still fails to cover whole exonic regions over here, and variants residing in these regions are completely missed. We've recognized these issues with the standard exome at Personalis, and we built an ACE exome designed it works by taking the standard exome here in blue and adding supplemental coverage in green. These are combined in an effort to maximize genomic content and to provide uniformity in coverage to increase confidence in calling variants. Not only do we target exonic content over here, but we also augment coverage of biomedically important regions outside the exon, such as regulatory regions, intergenic and intronic areas, as well as difficult regions of the genome like high GC content areas. We call this process our gene finishing metric. And using this approach, we achieve finishing of over 8,000 genes, more than 1,300 of which are actually cancer genes. This work was, of course, not trivial. It required to start with a sample, optimize different sample types and library preparations, add multiple parallel sequencing with custom probes that target difficult genomic areas, and we integrate all these steps to provide uniquely accurate data sets and high-quality results at the end. The results now, if we look at the STK11 gene example again, our augmented coverage with ACE, shown here in green, has supplemented the gaps in the gene to provide 99% coverage of exonic regions in a uniform manner. And we achieve this substantial augmentation for thousands of genes and enable access to information in these green regions. In this manner, clinically important variants residing in these regions that are completely missed with a standard exome would be detected confidently with our ACE supplementation. And here is just one example of a biomedically critical variant on the U2AF1 gene. U2AF1 is a component of the RNA splicing complex, the spliceosome. Mutations in this gene are very frequent in myeloid disorders, including myelodysplastic syndrome and acute myeloid leukemia. And specifically, one missense mutation identified affecting the serine at codon 34 is associated with an increased risk for reduced overall survival in myelodysplastic syndrome patients and shorter time to AML transformation. This specific variant resides in a region that is insufficiently covered by a standard exome, here in blue, and that has benefited substantially from ACE augmentation, the green region over here. On that same gene, you can also note the suboptimal coverage of the first exon with the standard exome and how it harbors multiple other mutations that would be missed with other solutions. I mentioned earlier that not only do we target the exon content, but we've also augmented coverage of biomedically important regions outside the exon, such as regulatory regions, intergenic and intronic areas. And here is an example that illustrates the value of this approach. TERT is one of many cancer genes that we augment with our ACE exome and cover on our cancer panel. And this is a content plot showing both exonic and intronic regions of that gene. An intronic variant was identified in this region, highlighted here in red, and is associated with increased risk of triple-negative breast cancer. Also, the TERT promoter mutations are associated with aggressive trait of different types of cancers. And here is just a snapshot of the number of mutations that occur in this area in the promoter region. Our ACE exome supplements the noticeable gaps in intronic and promoter regions to provide this complete genomic information from every gene. Here are some additional cancer genes with gaps missed by standard exomes and panels that we are able to achieve high levels of gene finishing and uniformity for. And really, I can show you pages and pages of these blue-green plots of the 8,000 biomedical genes that we finish. If you look specifically at cancer-related genes and the number of genes that we finish, Personalis finishes 20 to 30% more genes than other commercially available exomes on the market. We recently published on our patented augmented clinical grade exome in Genome Medicine. This work was co-authored with the National Institute of Standards and Technology and Genome and Bottle Consortium. In this paper, we detail how our approach enables more uniform coverage and higher sensitivity compared with other platforms, including whole genome sequencing. My colleague Heather Wetzel discussed this publication in greater details in a previous AMP webinar, but I will quickly share with you key figures from the paper. In this first figure here, we compared the finishing performance of ACE exome to that of 4 other commercial exome platforms, as well as a 30x PCR-free whole genome sequencing. And all of these platforms were sequenced at the 12 GB level. And in this graph, we're showing the percent of exons finished at 20x or higher depth for each platform tested. On the top left of the chart, it shows that ACE finishes 100% of exons with at least 90% covered at 20x depth. If we follow that green line to the right side of the plot, here we can see that ACE is consistently finishing a greater proportion of exons than all of the other platforms, including whole genome sequencing, with around 90% of all exons covered at greater than 20x read depth. As one example of how the unique enrichment approach used in ACE benefits tough to sequence elements like high and low GC content regions, we plotted percent finishing against GC content. ACE, here in green, the green line, maintains a high level of finishing at all GC content levels from 30 to 80%. And looking at the right side of the plot, we can see that we approach 80%, as we approach 80% GC content, most platforms have a substantial drop in performance. ACE, on the other hand, maintains a high level of finishing even at very high GC content levels. Based on this technology I just described, we've built cancer products to meet different needs in the research field. Our cancer portfolio includes ACE ImmunoID for immuno-oncology applications and an ACE extended cancer panel of 1,600 genes for DNA and RNA analysis. The ACE ImmunoID is really a singular solution that combines both DNA and RNA analysis to obtain the most comprehensive genomic and transcriptomic data for key immuno-oncology applications. It addresses the needs of studies investigating neoantigens, anti-cancer vaccines, immune checkpoint inhibitors, as well as other applications in immuno-oncology. And really, immuno-oncology is a prime example of a cancer research area that benefits from comprehensive tumor genomic characterization. Three main features to characterize in tumors are the mutational burden, neoantigen signatures, in addition to the need to assess immune checkpoint gene expression and enable the identification of targets for immune checkpoint blockade. Starting with the first feature, determining the overall mutational burden of a tumor has been beneficial as a major marker for immunotherapy response. In this study over here, if we look at the left plot, patients with durable clinical benefit on pembrolizumab, which is an anti-PD-1 therapeutic, showed substantially higher counts of non-synonymous variants as a group than patients with no durable benefit. Further, in the right plot over here, progression-free survival on anti-PD-1 therapy was significantly greater in patients with high mutational burden. These findings, as well as other similar studies, highlight the importance of using whole exome sequencing of tumors to determine mutational load and better evaluate patients who would benefit from immune checkpoint blockade. In order to profile tumors for mutational load, it is very critical to have an accurate assay and corresponding bioinformatic pipeline, of course, for somatic variant calling. With the ACE exome assay that I just described, we are able to achieve accuracy and uniformity in coverage for high confidence true somatic variant calls. And these sequencing plots here show ACE performance in green for PRDX5 gene and actin4 genes. PRDX5 in melanoma and actin4 in non-small cell lung cancer have been shown to carry somatic mutations that contribute to the mutational load in these types of cancers. And variants residing in green regions would be missed with standard offerings here in blue, leading to incomplete landscape assessment. On the other hand, the ACE exome fills in those gaps and gives a true representation of the genomic landscape for each cancer sample. We call small somatic variants as part of the ACE ImmunoID cancer analysis pipeline, and these here are just some examples of different cancer types that we analyzed with ACE exome and called small variants to get a sense for the mutational burden. And we used basal cell carcinoma as an example of high mutational burden and compared it to pheochromocytoma and renal cell carcinoma type of cancers. And as you see, the more the somatic variants, the higher the mutational burden, and therefore, the higher the likelihood of having a positive response to immunotherapy. Now, this type of data results from a tumor normal paired analysis that allowed us to obtain a true representation of the mutational landscape in each cancer type. Once the mutational burden is determined, it is also important to characterize these variants and identify the ones that are capable of eliciting an immune response against the tumor. These somatic variants can occur anywhere in the genome, but sometimes occur in a protein coding exon of a gene and can therefore be expressed in the RNA. Particularly when the somatic variants are missensed, this can result in the expression of a mutated proteins with alternate peptide sequences. The peptide sequence that contains the variant amino acid will then be presented in the MHC as a neoantigen, leading to an immune response when it is seen by the T cell receptor on the T cells. And this is the mechanism by which the immune system sees the cancer cells as a target for attack. These neoantigens are truly personalized cancer therapeutic targets. Every cancer patient will have a different set of these somatic mutations in their genome, resulting in a unique set of mutated proteins and therefore a unique set of neoantigens. In other words, every patient has his or her own personalized set of therapeutic targets in the form of neoantigens. One of the keys to personalized immunotherapy is identifying the unique set of neoantigens in each cancer patient that can be targeted. We have therefore created a sequencing platform built upon our ACE technology to identify candidate neoantigens from cancer samples. A cancer sample and a paired normal sample here from the same patient are sequenced with the ACE cancer exome, which is augmented to optimize coverage across over 8,000 medically relevant genes. This paired sequencing data is then analyzed through our tumor normal paired somatic analysis pipeline and will generate the set of all somatic variants that are predicted to cause a protein coding change that could ultimately result in a neoantigen. We then use RNA derived from the same tumor sample from which we got DNA to perform ACE transcriptome sequencing. Here, ACE transcriptome features the same augmented design as the exome, but targets the RNA instead. Somatic variants are called in the RNA as well, and then they are cross-referenced with the somatic calls that are called in the DNA. This gives us the set of all somatic variants that are actually expressed and therefore are most likely to serve as candidate neoantigens. Now, going back to our basal cell carcinoma example, based on the paired tumor normal somatic analysis with the ACE exome, this basal cell carcinoma had 222 somatic variants in the DNA. When sequencing the RNA with the ACE transcriptome, it revealed that 44 of those 222 variants are actually expressed by the tumor. This demonstrates how powerful a filter the RNA sequencing can truly be for the identification of potential neoantigens. It is also very important to mention that a good number of these expressed variants that we identified with RNA sequencing are located in coding regions that have benefited substantially from ACE augmentation. And this here is just one example of a somatic variant that is a missense mutation in the coding regions of the RPS2 gene. The mutation is represented by the red line in the middle of the tracks here in this figure. The light red tracks are the reads supporting this call in the DNA, and the blue tracks are the reads supporting this variant call at the RNA. And if you look at the sequencing plot below for the RPS2 gene, the expressed somatic variant is present in a region that is dramatically improved by ACE augmentation, here the green region. And this demonstrates the increased sensitivity of the ACE exome and transcriptome in guiding the detection of potential neoantigen. What is different about our ACE transcriptome solution? You know that many clinical studies depend on tissue archives that have been fixed using formalin-fixed paraffin-embedded procedures, FFPE. This process makes it difficult to obtain a pure sample and also leads to RNA degradation. In the past, enrichment methods such as poly-A selection have shown certain limitations with these types of samples. Also, ribosomal depletion methods suffer from inconsistent ribosomal RNA removal and a high percentage of reads mapping to intronic and intergenic regions, especially for FFPE samples due to cross-linking. So Personalis has developed an exome capture transcriptome protocol based on our ACE technology and on our enhanced ACE probes to overcome these limitations and can produce high-quality transcriptome results from FFPE tumor samples. The ACE cancer transcriptome enrichment protocol directly selects for transcripts using complementary capture probes, eliminating as such background and focusing the sequencing on regions of high interest. We also developed a DNA-RNA co-purification method without affecting the DNA yield, and this is especially important with limited tumor samples. With this in mind, when comparing the performance of our ACE cancer transcriptome to ribosomal RNA depletion methods, you can clearly note the difference in the percent of bases that had mapped within the coding and UTR regions of the RNA. Sequencing using the ACE transcriptome protocol demonstrates that greater than 95% of the bases are aligned to regions of interest, hence increasing genomic information obtained from each sample. And just as I showed you the exome blue-green plots, also our RNA sequencing benefits from the coverage and accuracy improvements that we discussed earlier. Here are some RNA coverage plots for 4 different cancer genes, and you can see the gaps that are filled in nicely with the ACE that would be missed with standard protocols. This brings us to the third feature in immuno-oncology that is important to investigate, and that is the expression of immune checkpoints and their ligands as therapeutic marker for immune checkpoint blockade. We know that increased expression of these receptors and ligands has been strongly associated with positive response to therapy. And in these figures here, these figures show that the group of patients who respond to PD-1 therapy is enriched for PD-L1 expression, demonstrated here with the brown color by immunohistochemistry. And in the bar chart, we can see in gray that patients who showed an objective response to PD-1 blockade also showed positive expression of PD-L1 ligand. This means PD-L1 expression is a therapeutic indicator for increased response to PD-1 blockade. With the ACE cancer transcriptome assay that we offer, we are able to accurately determine gene expression of some of the most studied immune checkpoints, for example, PD-1 and PD-L1, for which there are multiple drugs under investigation, but also our assay would provide the foundation to explore other immunomodulatory molecules that could influence response to immune checkpoint blockades and help advance the field of immuno-oncology with these new findings. We evaluated the performance of our ACE cancer transcriptome in determining immune checkpoints expression. Gene expression here can be clearly seen in these different patient-derived cancer samples that I described earlier. All of these samples were sequenced on the ACE and analyzed using the Personalis cancer analysis pipeline. The expression in transcript per million of the immune checkpoint genes is elevated quite dramatically compared to the expression in the control sample, which is here a lab-grown cell line. Moreover, we can also see that the expression of these genes varies from one cancer type to another. For example, the basal cell carcinoma has substantially higher expression of PD-1, PD-L1, and PD-L2 than either of the other 2 tumors. This suggests that the patient with basal cell carcinoma may respond more strongly to anti-PD-1 immunotherapy. And for every RNA sequencing run we perform, we return data of this format to facilitate exactly this type of research and analysis. In conclusion, for this part, immuno-oncology represents a major area of personalized medicine. When studying immunotherapy, we need to be able to assess the expression of immune checkpoint genes, the mutational burden of the tumor, and the identity of neoantigens. Exome and transcriptome sequencing enable us to assess all of these areas, as well as additional therapeutically relevant cancer variations. And Personalis ACE augmentation has a tangible benefit for all of these applications because it enables more comprehensive assessment of these genomic elements. With the ACE ImmunoID assay, we can return a wide range of analytics from raw files to filtered reports for both DNA and RNA to allow for a more comprehensive profiling and characterization of each tumor sample. So now for a more focused profiling of tumor samples, we developed an extended cancer panel of 1,600 genes to allow tumor profiling for clinical trials or even deeper interrogation of low allele frequency mutations in cancer, in addition to other applications in cancer research. And in the next few slides, I will describe the design of our cancer panel as well as some of its specifics. So to offer the most comprehensive end-to-end solution for cancer profiling, in addition to our ACE whole exome and whole transcriptome, we built the ACE extended cancer panels for tumor DNA and RNA analysis that delivers high depth and accurate sequencing on a larger set of cancer genes. And when we started the design of our assays, we looked to make sure we had complete coverage over genes with clinical utility, consensus driver genes, key cancer pathway genes, as well as pharmacogenomics and immuno-oncology genes. And this resulted in over 1,300 cancer-related genes, which we then specifically augmented on our ACE exome and became the basis for our ACE cancer panel, which is the largest cancer panel of its kind. With the panel, we aim to sequence at a depth higher than 500x to be able to detect those minor allele frequency mutations that are below 5% with greater sensitivity. In addition to DNA analysis, we built a complementary transcriptome and panel for RNA analysis. And these assays also use the ACE technology as a foundation and allow for better RNA finishing, the detection of known and novel fusion events, as well as gene expression analysis and expressed variant detection. The cancer content we target with the ACE cancer panel includes more complete coverage of key cancer pathways, giving a more comprehensive view of the tumor biology. The numbers here in green represent the cancer genes on our panel compared to another standard panel here in gray. And if you look at the numbers, we augment significantly more genes for each signaling pathway involved in cancer than the standard panel. And this allows a broader view of the networks in cancer and a better understanding of the tumor biology. If we categorize these genes by cancer type, you can see that the panel covers both solid and hematological cancers. We include all genes you would find on standard panels here in gray, but we've also augmented the content here in green and target more genes by cancer type to allow for broader analysis. Now, in the next few slides, I will discuss some case studies where Personalis is able to provide the most comprehensive coverage of genes through using the ACE panel. In this first study, we analyzed 5 FFPE samples from patients with aggressive form of glioblastoma. These patients had a late-stage disease, stage 4. 2 patients had EGFR-V3 mutation, all had EGFR amplification and PTEN loss, and no evidence of IDH1 mutation. Now we know that EGFR mutations are very well characterized in glioblastoma, and especially EGFR-V3 is known to confer resistance to tyrosine kinase inhibitors due to exon skipping that results in a truncated form of the receptor. The ability to detect this type of mutation is important for personalized cancer therapy, and using the ACE extended cancer panel for DNA and RNA analysis allowed us to accurately detect and identify the EGFR-V3 mutations in these samples. This graph here shows the number of reads mapping to exon 1 and exon 8 in EGFR, and also it provides the evidence of exon skipping from 2 to 7 in these 2 patients who are positive for the EGFR-V3 mutation. In addition, by analyzing both the DNA and the RNA, we can correlate copy number alterations with gene expression. And in this plot, we show evidence of changes in copy number by chromosome supporting EGFR amplification over here, chromosome 10 loss leading to P10 deletion in these patients, but also other hallmark alterations in glioblastoma. And this also demonstrates the feasibility of using FFPE samples with the panel assay that we developed. Our RNA panel is also built on the same ACE technology and accuracy, and it's complementary to the DNA panel and includes 1,300 cancer genes important in cancer biology. For the RNA analysis, we exclude intronic RNA from unspliced transcripts, and this to allow elimination of background, but also deeper interrogation of messenger RNA. And as I mentioned earlier, we've optimized our protocols to extract RNA from FFPE samples, even if the RNA is slightly degraded. This application allows for the detection of known cancer fusion events, but also the identification of novel fusions in cancer for each sample. And here are some examples of a well-characterized fusion event or fusion events in CML, non-small cell lung cancer, and ovarian cancer. And these fusions are here represented in those circles plots that we were able to accurately detect using the cancer panel for RNA analysis. This here is a complete list of deliverables we returned for DNA and RNA cancer panels. In addition to raw files, we also generate somatic variant analysis files and various plots and statistical summaries for both the DNA and the RNA for a more comprehensive reporting for each sample. And to conclude, Personalis has addressed specific challenges in sequencing and analyzing tumors. We offer solutions to detect low-frequency mutations, especially from heterogeneous tumors and tumors with variable purities. We are able to handle different sample types, and we've optimized our sample preparation protocols to simultaneously extract DNA and RNA from each tumor sample. We've addressed gaps and inaccuracies in sequencing and have built a clinical-grade genomic solution with uniform and augmented coverage of biomolecularly important genes. In addition to developing comprehensive assays such as our whole ACE exome and transcriptome and our extended cancer panel for DNA and RNA analysis to meet clinical trial and research needs in the we will be at AMP in Austin, Texas from November 4 to 7, so please join our workshop and stop by our booth to learn more about Personalis and meet our team. I'd like to thank you all very much for listening today, and I'm happy to take some questions.
Okay, excellent. Thank you so much, Christelle. So, all right, ladies and gentlemen, now we'd like to open up the question and answer session here of our webinar. So if you haven't done so already, you can go ahead and answer some questions, or actually type some questions into the control panel there and send them our way, and we'll get those answered for you. So I've received a few questions here. So Christelle, the first question that we have out there is, are your assays configured to run when a matched normal sample is not available?
Yeah, this is a very great question. We actually have two different somatic variant calling pipelines, one for when the paired normal sample is available, and one when the tumor is being analyzed without a paired normal sample. And in the tumor normal paired analysis, the normal sample is not used simply to subtract variants from the tumor variant set. Instead, the tumor and the normal are processed together simultaneously to increase accuracy of somatic variant calls. And because this approach is so effective at identifying true somatic variants, we utilize a downstream filtering process that is catered specifically for tumor normal somatic variant calls. And when the normal sample is not available, and we understand it is the case for many studies, we utilize a substantially different somatic calling approach. And if you do the bioinformatics correctly, you could actually get accurate results with a tumor-only mode. Our approach includes use of a panel of normal samples to reduce the chances of false somatic variant calls. And naturally, when calling somatic variants without a paired normal sample, germline variants can be difficult to differentiate from somatic variants. And for this reason, we use a different set of variant filters with our tumor-only somatic variants that increases the specificity at low variant allele frequencies.
Okay. Excellent, thanks. All right, the second question that we have here from the audience is how much material is needed for your assay? How do you also assess sample quality?
Great, yeah. So we, as I mentioned during the presentation, we are able to handle different sample types, and each have different requirements. For example, for FFPE samples, we can work with 4 sections, 5 to 10 micron thick. We also have the capabilities in-house to process FFPE blocks and handle them in-house. For fresh frozen tissue, for example, we can work with 25 to 30 milligrams. We're also able to work with blood samples, peripheral blood mononuclear cells, bone marrow aspirates, and we would ask for about 1 million cells with these types of samples. We also accept genomic DNA and total RNA, and we would ask for about 250 to 500 nanograms. And of course, these are desired amounts. We have certainly worked with less material, and it really depends on the quality of the tumor sample. So that said, each sample that we receive goes through a robust quality control process, and Personalis assessment of samples involves initial qubit and nanodrop readings, followed by Agilent tape station for highly reproducible and automated quality assessment using gel sizing. We also perform a TaqMan assay, and this step helps us determine enzyme accessibility, but also helps us determine how much input material to use for sequencing. And we always make sure to communicate these results to our customers before we proceed with sequencing.
Mm-hmm. Great, thanks. And kind of along the same lines, one of the specific questions here was what amount of tumor content do you tend to request or observe with your assay?
Right, so we would like to work with tumor content that is greater than 20 to 30% especially from FFPE samples.
Okay, great. Another question out there is how is the data delivered?
Yeah, so we deliver the data on a hard drive, and the drive would include all the raw files in addition to quality control reports, summary statistics files, annotation and analysis reports, and we can also encrypt the data if needed.
Okay, great. What sample type is tend to use for the normal sample in your ACE assay?
We use a whole blood type as a normal type, but we also have a proxy normal database that we've developed and established at Personalis, and that's unique to Personalis, using a panel of normal samples.
Okay, excellent. Thank you. So thank you so much, Christelle. We've now come to the end of our session today. Thank you very much for everyone who has joined us. We hope you all found this presentation to be beneficial. And again, a big thanks to Christelle for leading this informative and interesting discussion.